and welcome back to Creation Myths. Today we are going to be looking at another one of the three post-debate damage control articles written by Dr. Jerry Bergman following our debate on the topic Common Descent versus Common Design from January 18th, 2025. In that debate, during the question and answer session at the end, I got a great question about observed examples of evolution, and I gave a few of my favorites in response. I'm going to show you just a short part of that bit right now, because that's the context for everything we're going to be talking about here. So watch this little bit, and then we'll talk about it.
Question for Dan. What is the best experiment you can point to for the most substantial macro evolution? Not just homology and comparisons, but actual experiments. Thanks. Thank you.
Oh man, I love this question. There's so many good ones. So one of my favorite ones is the evolution of de novo multicellularity in a species of green algae that has no multicellularity in its evolutionary history. And over the course of about 2 years in the lab, they evolved into an obligatory multicellular state where they had no unicellular stages in their life cycle. So that was the de novo evolution of multicellularity. And it wasn't just that individual cells accumulate in a colony, it was that a single cell, they divide and they stick together and they form a multicellular structure just like, we do. So that's a really good one. This is the paper I was referring to in that short segment when I talked about the observed evolution of multicellularity in green algae. It's called De Novo Origins of Multicellularity in Response to Predation by Heron et al. 2019. And I love this paper because it is a direct experimental observation of a major evolutionary transition, the transition from unicellularity, having just one cell, to multicellularity, having multiple cells by necessity. Here's the article Dr. Bergman wrote to clean up that moment from the debate, titled The Debate Between Dr. Jerry Bergman versus Dr. Dan Stern-Cardinal, Common Design or Common Descent, Single Cell to Multicellular Evolution Observed in Algae. Let me just say before we go any further, last time I addressed one of Dr. Bergman's responses, I went through the paper line by line, but I got some feedback that was more tedious than it needed to be. So, fair enough. We're not going to do that for this article. We're not going to go line by line. But of course, I've linked the article down in the description, so you can read the whole thing if you want. But I'm going to pull out excerpts and talk about the highlights as we go through. So the article starts with a part getting my exact description of the evolution in question wrong. That's the video clip that you just watched, and this part misquotes me fairly substantially, but that's not really the point. But I do want to make clear the quote attributed to me here is not exactly fully correct. You can go back to the transcript of the debate. You can go back just a minute and watch the clip if you want to confirm that these words are not exactly what I said. It's a little bit muddled, and it kind of mangles the point a little bit, which is why I point it out. There's a few things here, like talking about that it's a new species, for example, that I didn't say, and I don't want to be on the record as that being part of my claim, right? My claim here is pretty narrow, that we have a directly observed example of multicellularity evolving from a single-celled ancestor in the lab. That's really it. In the next paragraph, Dr. Bergman quotes the author of the paper I cited in which the de novo multicellularity was reported, and I want to highlight this line. Quote, "The transition from unicellular to multicellular life was one of a few major events in the history of life that created new opportunities for more complex biological systems to evolve." And on this, I'll just say yes, I think in general that statement is accurate. Multicellularity is a major development that can facilitate the evolution of subsequent complexity in a lineage. And one reason this paper is so important is that it shows how that can happen relatively quickly. Now we're getting into the part of the article where we're going to see a bunch of either errors or misdirection or obfuscation from Dr. Bergman, and I should note his co-author, none other than Donnie Badinsky from Standing for Truth. And this first one here is very simple. They reference a 2021 paper on multicellularity saying, quote, "Clumping together of individual cells does not meet the definition of multicellularity." Correct. That's colonialism, not multicellularity. When you have multiple individual cells that are doing their own thing and then they aggregate together, you're right, that's not really usually considered true multicellularity. And that's also irrelevant because that's not the observation I was talking about in the debate. It is something that was observed in this experiment, but it's not the specific finding I care about, right? It's not the thing I'm pointing out as an observed example of the evolution of multicellularity. And I want to point out that Dr. Bergman and Mr. Badinsky are not the first creationists to make this error, and I'll be generous and I'll call it an error rather than a misrepresentation when it comes to this paper. There's another creationist with a blog, Dr. Weil, who years ago made this exact same thing, consistently referring to the observation in this paper as teaming up or aggregation, when what we're actually observing is a multicellular structure growing via cell division, via a process called mitosis, where one cell divides into 2, divides into 4, 8, and so on. And those cells, when they divide, they stay together as a cohesive multicellular structure. That's the key observation that we're talking about here. A little further down, they write, does the article Dan used to document his claim that de novo origins of multicellularity have been documented to occur in one generation hold up? And I just want to point out here that this did not happen in one generation. If you go back to the original paper, I didn't say it was in one generation, and the paper doesn't say it was one generation. In the debate, I said 2 years, but I actually got that wrong. If you go back to the original paper, it was actually 50 weeks, which translates to about 750 generations in this particular species of green algae, not one generation. In this next section, there's a pretty egregious bit of equivocation that I want to point out. They write, the article's example meets none of the conditions required to evolve from single-celled existence to the multicellularity existing in higher-level organisms. Did you catch the goalpost move there? Now we're talking about, quote, the multicellularity existing in higher-level organisms. Do you think that the earliest, most ancestral forms of multicellularity, the kind that could evolve experimentally in the span of a single year, would be comparable to higher-level multicellularity? That would involve things like tissues, organs, and organ systems, things we tend to see associated with animals and plants. That's not the only kind of multicellularity that exists. There are a lot of multicellular lineages, multiple types of red and green algae, plants, animals, fungi, also brown algae, golden algae, and depending on how you define it, even some amoebozoa like slime molds. Many of those have no more higher-level organization than what's observed in this experiment. The fact that Dr. Bergman and Mr. Badinsky even included that caveat tells you everything you need to know. We could basically end this right here on the basis of that line. They know multicellularity evolved in this experiment, so they have to push the standard back to a more derived, more complex form of multicellularity. But in doing so, they effectively concede that an earlier, more ancestral form of multicellularity did in fact involve experimentally in this experiment. And also I'll highlight again, the line right below that mentions clumping, and again, that's not what we're talking about. So there are a whole bunch of errors here, right, in terms of what they're arguing against versus what actually happened and what I'm pointing out from this paper. A little bit further down, they wrote, quote, in this case, the presence of a predator triggered what appears to be a pre-existing ability that allows algae to clump, which protects it from predators. No. The observed multicellularity was not pre-existing. These algae do have the ability to cluster together in response to predation. They do not, in nature, have the ability to be multicellular, as opposed to colonial, all the time. Again, in the wild, they can team up, you might say, for defense. What I'm talking about is how they form multicellular structures via mitotic cell division, where the daughter cells stay together, resulting in a single multicellular individual. These two things are not the same. They're not the same mechanism, even though the multicellular structures that result might superficially resemble each other. The thing here is that Dr. Bergman and Mr. Badinsky are not addressing the actual argument I made, regarding the clonally generated multicellular structures that grow via mitosis and cytokinesis and then stay together following that process. That's not present in the ancestral organism. To understand this transition in a little more detail, let's look at some phylogenetics of the group that contains green algae called Archaeplastida. This is one of the eukaryotic supergroups, we call them, which is a level in the hierarchical organization of life between domain eukarya and all the kingdoms of eukaryotes like plants, fungi, and animals. Okay, so within this group called Archaeplastida, we have the red algae, we have several lineages of green algae, and we also have the plants. Chlamydomonas, the group that's in this paper, is in the chlorophyte lineage, which you can see right there below the red algae. That lineage does contain some species with multicellularity, but if we zoom in, we can see how that isn't the case for Chlamydomonas themselves. So let's zoom into a more detailed phylogeny of their specific lineage. This figure is from a 2016 paper called Alternative Evolution of a Spheroidal Colony in Volvocene Algae, Developmental Analysis of Embryogenesis in name of the group of organisms they're talking about. You can see here that the most derived members of this lineage, all the way on the right, are green algae called volvox. And I just want to say volvox are super cool, and I love volvox. And if you live in the right area, like where I live, you can go to like a pond or a river and scoop up some water, and you might find some volvox in there. It's super cool. You look at them under a microscope. Sometimes the colonies are big enough, you can even see them with a magnifying glass. It's super cool. That doesn't matter. That's just an aside, something that I really like. Anyway, this thing called volvox, these spherical colonies of green algae, they actually have a fairly sophisticated form of multicellularity. It's actually improper for me to call it a colony, sort of, because they have fairly robust cell specialization, where in some examples, they actually have coordinated direction of the movement of the group being directed by certain cells that have pigmented spots that can detect sunlight, so they can optimally move for photosynthesis. And then other specific cells within that spherical group, we can call it a colony, whatever, within that spherical structure are responsible for reproduction, right? So they have a fairly sophisticated form of multicellularity there. But all the way at the other end of the phylogeny, all the way to the left by the root of the tree, we have the Chlamydomonas. It has the ancestral traits when it comes to multicellularity, which is to say it's unicellular. They exist as single cells. In fact, the suffix in its name, monas, in Chlamydomonas, that means solitary, highlighting how they're unicellular. But then under selection to avoid predation or ward off predation, they become obligately multicellular over the course of about a year, as reported in the paper I've been talking about. They follow a similar trajectory as the other members of this clade, where, as you can see from this figure, multicellularity has evolved 2 separate times. So again, I want to be clear, the point I'm making by kind of going down this rabbit hole with the evolution of Chlamydomonas and its close relatives is that Chlamydomonas itself, the model organism used in this paper, does not, in its evolutionary history, have multicellularity. It has the ability of individual cells to clump together to form colonies, which is a different mechanism from the mechanism observed in the 2019 paper that I was referencing during the debate. And all that matters because here we have a main theme of the second half of this article. Quote, "These organisms may have been able to move between free living and clumping, but lost this ability due to deleterious mutations. As a consequence, they are now stuck in the clumping mode." This is just the idea that the multicellularity, the incorrect version that Dr. Bergman and Mr. Bedinsky are portraying as clumping, is an ancestral trait, and the ability to do anything else has been lost. Now first, the clumping is an ancestral trait. It's important to note that. But it isn't the multicellularity that the authors describe and the multicellularity that I'm pointing out as the important evolutionary advance that's being directly observed here. Again, the novel multicellularity involves a different mechanism, one that the ancestral Chlamydomonas population did not possess, as I showed, as we went through those phylogenetic trees. That was the point of going through the phylogenies. And we can end here with this line, quote, "This case does not support the evolution of multicellularity required in higher animals for many reasons." We can again point out the bait and switch between the most ancestral form of multicellularity, which is documented here, and the more derived form involving tissues, organs, and organ systems that Dr. Bergman and Mr. Budinsky want to talk about. I'll again point out that the fact they're moving the goalposts means they know that if we talk about multicellularity as a whole without further qualification, the ball is in the net. So they have to move the goal so they can pretend the evolution didn't happen. But the very fact that they had to do it tells you they know they're wrong and they can't win the fight on the grounds of what's actually being observed in that paper. The big picture I want to leave everyone with is that in less than a year, a population with no multicellularity in its evolutionary history was shown to evolve obligate multicellularity, not via the aggregation of separate free-living cells, but through mitotic growth. We witnessed this process happening. What had to cause it? There had to be some underlying genetic changes that led to the transition. We know that's the case. Creationists love to look at something like this and say, well, it might just be epigenetics, as though that's a magic wand they can wave to negate any observed evolutionary change they want to be able to pretend didn't actually happen. Now, first, you can't just say that without evidence, right? Like, if you're going to claim there's a mechanism underlying this observation, you need to provide evidence that that's actually the case. But second, the whole point of epigenetics is that it's responsive to environmental conditions. And in the wild type, in the natural populations of this algae, when you remove exposure to the predator, they stop clumping together, right? It's a facultative trait. But in this case, this multicellularity persisted even once the predator that provided the initial selective pressure was removed. It was no longer facultative. It was obligate. If it was just epigenetic, the algae would have reverted back. So in this case, even if the proximate cause was just epigenetic regulation of gene expression, changes to epigenetic regulation that resulted in obligate multicellularity as opposed to a facultative plastic trait, those changes must have occurred through a different mechanism, have to be mediated by changes to the underlying enzymes. So at the end of the day, creationists, by saying this is just epigenetic, as long as we all agree that was obligate, it wasn't a reversible phenotypic plasticity kind of thing, you're just acknowledging the same mechanism, right? Mutations leading to a change in phenotype that was then selected for, you're just adding an intermediate regulatory step. It's mediated through epigenetic mechanisms rather than, direct changes of the genes involved themselves. So if we go back to this figure that shows very clearly what happened to this experiment, that's how we know algae evolved de novo true multicellularity. And it's one of my favorite examples of a major evolutionary transition that has been directly observed. Thank you for watching. Please remember to hit like, leave a comment, subscribe if you're not already subscribed, and consider becoming a channel member because you'll get access to pre-recorded videos like this right away without having to wait for the public release. Thank you all again for watching. Don't get fooled.